All right, so let's quickly go through these guys so we can get to the lab. Number 1, purple stuff with the dots, okay? That is the rough ER, okay? Okay, and the rough ER's job, transport proteins. 1 mark each. All right, number 2 is the smooth ER. Smooth ER will accept any one of the following. Okay, makes hormones, detoxifies poisons, breaks down carbohydrates, and makes lipids. Okay, any one of those is fine. Number 3, okay, is the nucleus. Okay, nucleus stores DNA. Number 4 is the nucleolus. Okay, and the nucleolus copies DNA, controls protein synthesis, okay, makes RNA. I'll accept any one of those. Number 5, the outside of the nucleus is the nuclear membrane. Okay, and we'll say controls what gets in and out of the nucleus. Okay, we can't really say endo and exocytosis because cytosis implies the cell as a whole, but if they have that written down, I'll accept it this time. Okay. All right, the little dots for number 6 are the ribosomes, and they make protein. Number 7 is the Golgi apparatus. Okay, and the Golgi apparatus controls exocytosis, or packages wastes for excretion, or gets rid of wastes. Something about waste disposal would be fine. Okay. For number 8, that's the cell membrane. And for that one, we do have to have endocytosis and exocytosis. Dom, question? No, the animal cell does not have a cell wall. Okay, it's only cell membrane. All right, number 9 is your mitochondria. Okay, burns sugar for energy. Burns glucose for energy, if they have that. All right, number 10 is pointing at all these little bubbles with the stuff in them. Okay, those are lysosomes. I would also accept peroxisomes if they wrote that. Okay, lysosomes digest food, break down food. Okay, release energy from food, something about food. Okay. Number 11, okay, number 11 is pointing here at these things that are circled. Those are the centrioles. Okay, the centrioles help with cell division, or they grab DNA and pull it to either side of the cell, or something along those lines would be fine. And number 12, the stringy things. I will accept any of these as the answer for stringy things. Okay, I will accept microfibers, microfilaments, or microtubules. They all have the same job. They help support or keep the shape of the cell. Okay. Give them a mark out of 24. Let them see it. Put it in the box. Dom, question? No. Nope. All right, so get them a mark out of 24 there, guys, and then back to them. All right, so cell anatomy lab here, guys. So today, when we go into the lab, going to be using the microscopes again. However, they have been put away in the cupboards. They're not hard to find, okay? The same setup will apply, though. I will have all the slides you need up at the front of the room. You'll be looking at 4 different things today, so hopefully you're a little better at finding stuff than you were the other day when we started, okay? And you'll be able to find these things fairly quickly, okay? What we're looking at here, this time we're looking at confirmation of some stuff about the cell theory, okay? One of the big things is that cells carry out the basic functions of an organism, okay? We've said that that's going to come up over and over again in this unit. It's huge for this lab. Probably going to be part of several of your answers, okay, in the analysis, but it's a huge part here, okay, of this particular lab. So, the problem is cells must carry out the basic functions of an organism in order to survive. Thus, they must have structures within their cells that carry out these basic functions. So essentially, what we're looking for here is common structures between the cells that we're going to look at. Okay, are you going to see everything that's in the cell? No, but you'll see a fair amount of it. Okay, you're not going to see ribosomes, and you're probably not going to see like really small stuff, but you'll see a few things for sure. All right, so be on the lookout for those. And sometimes, yeah, you have to use your imagination a little bit, but most of the time it's there. If you're not sure what you're looking at, call me over, and I'll have a look at it. Okay, so the design here: identify your manipulated, responding, and controlled variables, and explain your choices. Okay, are we looking at different things under the microscope today? Yes. Okay, some of them are animal, some of them are plant. Okay, some of them are, well, nothing in between really. They're either animal or plant. Are we looking at the two major types of cells? Okay, are we comparing the two major types of cells? Is that what we're manipulating? Okay, responding variable. What are we looking for here? Okay, we're looking at these cells and we're trying to find out what's in them, and most importantly, what things are in there carrying out the basic functions. Okay, so something to think about there. Controlled variables. One of the big ones here, guys, is these are different kinds of cells in that they are plant and animal, but they are the same type of cell. And we talked about two types. Okay, that part I'm leaving to you to look in your lesson 5 notes to find. Okay, one of the controlled variables is the type of cell we're looking at, and you need to find out what type it is we're looking at. Okay. Other controlled variables, okay, they have to do with what you're doing in the lab. Okay, basically, you know, are you looking at everything in the same way, same magnification, same technique, that kind of stuff. Explain what effect that would have. Make sure you explain your variables. Okay, hypothesis. Generate a hypothesis about which organelles will be present in cells and visible under our light microscopes. So this part, okay, which organelles will be present, that's probably which part of the hypothesis? Okay, we're investigating the cell theory here, all right? And that is what cells do. That would probably be the if part, what cells do. Perhaps cells do something like this. Okay, the and part, you talk about the experiment and what we're looking at. The then part should probably talk about what you think you're going to see. Okay, and be specific when you tell me what you think you're going to see. Don't just say, we'll see the parts. Because I'll see your two. Okay? Make sure you are specific about what we are looking at. Okay? For the procedure, okay, straightforward. Like I said, we're going to go in the lab. We're going to look at these various things. You are going to be making a wet mount today, and it is going to be of your own cells. Okay? From your cheek. We used to use a toothpick for this. We're not going to use a toothpick anymore. We got smart. I had too many people who were drawing blood with the toothpick. Yeah. I don't want you to look at blood cells that could contain disease-carrying stuff like hepatitis, HIV. Yeah. Not that I think any of you have that, but we can't take any chances. We're not allowed to have bodily fluids of any kind in the lab. Okay? No blood or any other bodily fluids, please, in the lab. Okay? None of those. So, saliva is not going to count as a bodily fluid. You're going to get a little bit of that on the end of the Q-tip. We're going to go CSI in there. Okay? You know how in CSI they always swab the inside of people's cheeks? The reason they do that, the cells in the inside of your cheek come off really, really easy. Okay? And so all you have to do is kind of put the cotton swab in there, rub it up and down, and twist it a little bit while you do that, and it'll be covered with your cells. And some of the stuff you ate. Okay? So you'll smear that onto the slide. Okay, just smear it around a whole bunch of times while you're twisting it, and then put a drop of water on there, put the cover slip on, and you're going to have lots of your own cells to look at on there. Some of them will be destroyed, some of them won't. If it's really big and dark, it's something you ate and not one of your cells. Okay? Generally, that's the rule here. If it's big and dark, it isn't yours. Okay? All right. For the analysis part, there's only one question here, guys. So I'm looking for a good, thorough answer, good explanation here. Based on your knowledge of cell structure and your observations of the cells we looked at today, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? All right, so we also got to think, we're looking at plants and animals here. They got some differences. What were they? Okay, explain your reasoning. And then conclusion and evaluation, same as always. All right. Questions on what we're doing there in terms of the report part? Everybody's good with that? Okay, again, make sure this answer is thorough. If all you write down is, I saw the nucleus, yay for me. 1. Okay, I want more than that. Okay, these are the things you're going to be looking at. Okay, so it gives you a rough idea of what to look for under the microscope. The plant cell you're going to be looking at, okay, on the slide it's going to say monocot, dicot, cross section. Okay, you need to look at this, and this is what you're going to see. Okay, these are the layers of the leaf, which we'll actually talk about in about another week or so. You want to look at this layer of cells here. Okay, we call this the Palisade layer. The cells are very fixed in their shape. Okay, so they're much easier to see. You can see a lot of the structures a lot better. Okay, things like that. So, okay, when you're looking at this, obviously the nucleus stands out there. Okay, you've got, you know, certain shape so you can tell where the cell is and where it begins and where it ends. All right, you can see a fair amount of structures inside as well. Again, you look at your cell diagram and you figure out what you think you're looking at. There's not really a right or wrong here, okay? I mean, when I look at your diagram, I'll see what you've done and what you've labeled. Okay, so like I say, do your best with the labeling. Obviously, the obvious structures I'm going to be picky about. Okay, the ones that you maybe had to extrapolate a little bit, I'll be a little bit more forgiving. Okay, unless you say that the little tiny faded circle was the nucleus, and then of course, okay, like if you're saying that right there is the nucleus, that's wrong. All right. The animal cell you're going to be looking at is from on the slide. It'll say generalized animal cell, or it'll say stratified squamous epithelium. That's the fancy way of saying, you know, when you scraped yourself and you got that red skin that doesn't quite bleed that's showing? That's what that is. Okay, so it's the layer of skin under the outer layer of skin. All right, that's what you'll be looking at there. Now, to give you some idea, okay, this here is a cell. One single one. Okay, how many? Okay, when you're doing your lab drawing here, you can do the same thing. It's going to be a little tougher when you cut that out. Okay, in paint, you got to isolate one cell and then blow it up nice and big. Okay, so you can see all the parts really well. Okay, when you're doing your lab drawing. Okay, but each and every one of these little dark dots is a nucleus. All right, it helps you to kind of find where the cell is. On your slides, the nuclei will be a little more purple than the rest of the cell. So look for little purple dots and then try and find the cell that's surrounding it when you're doing your lab drawing. Okay, everybody follow me there? All right, so that's your generalized animal cell. You're also going to be looking at blood, human blood that is preserved and sealed with contact cement underneath so you can't get anything from it. All right, in there, okay, you'll see the red blood cells. They're the kind of hazy pink ones. Okay, you'll also probably see some leukocytes, which are white blood cells. That's these funny looking ones. Okay, I just want you to draw the red blood cells. Now, a couple of tips on the red blood cell. Red blood cells are non-nucleated. That means they do not have a nucleus. Okay, they are rare in your body, but they do not have a nucleus. You will not be able to label very much on the red blood cell. As a result, the red blood cell should not be included in your analysis. Okay, so when you're saying, I saw these structures in every cell I looked at, don't include the red blood cell, because then you'll have a cell that didn't have a nucleus, and you might arrive at the conclusion that nucleuses aren't important. And they are. Okay, it's just that red blood cells don't have them to increase their surface area to carry blood. Okay, they're produced by your bone marrow. They don't work the same way as your other cells do. Okay, so you're just looking at them to have another cell to look at, because everyone always wants to know what blood looks like. So there you go. Okay, this is what your cheek cells will look like. All right, because it's not stained, it's not going to be bright and colorful like the other ones are. All right, so it's going to be darker. It's going to be gray. Okay, and each one of these things here is a cheek cell. All right, and you can see quite clearly several of the structures. In fact, it's probably in the cheek cell where you'll see the most organelles. All right, I have had groups in the past who have showed me their cell through their microscope and said, is this the Golgi? Is this the rough ER? Okay, is this a mitochondria? And I have actually been able to look at their cell and say unequivocally, yes. Okay, your cheek cells are a little bit bigger, and you can see a lot of stuff in them. Okay, even in the stratified squamous epithelium, this generalized animal cell, you can sometimes see a fair amount of things. You just have to look really close. Okay, and in the plant cell, obviously they're quite large as well, and you should be able to see a fair amount of detail in them also. Okay. One other tip with the cheek cell. You may need to adjust your lighting to see them, to make them stand out, and not just the brightness on the little rheostat, okay, but that that little tab that's underneath the stage that a few of you were kind of playing with the other day, okay? When you close that off, it will increase the contrast of the slide. That means make light and dark stand out more, and the cell will literally jump out, okay, at you when you do that. So when you close it off, if you're having a lot of trouble seeing it, close that off and you'll really see the contrast pick up, and it'll be a lot easier to see the cells then. Those cheek cells will be alive, yes. You won't see much. They don't do much. They just sit there. Devin, question? These are various. They're various powers. Plus, they've been enlarged on the screen here, so their magnifications are. This is about what you'll see. Okay, it's close. All right, the blood cells would be a lot smaller than this. That'd be the only one that would be a lot different. The blood cells would be a lot smaller. Yes, you are still taking picture of these and doing the lab drawings in the same way you're doing the ones from last time. Just make sure you only show me one cell or as close to one cell as you can when you're cutting those out. Okay? Everyone follow me there? Questions before we go across there, Alex? I'll decide a little bit later on. Probably I'm thinking like around the 8th of April, maybe something like that. Yeah, that's the rheostat. That dims the light source, but the ocular lens, the thing with the tab underneath, closes off a diaphragm like the iris of your eye, and that increases the contrast. Okay, so that's what I was talking about. You can use both, but you'll probably use the condenser diaphragm more. Okay, any other questions? All right, let's head across to the biolab then.